Oh, my stomach. Hungry again? When we consume carbohydrates, our digestive system breaks them down into glucose, which becomes fuel for cells. This extra energy is converted into glycogen.
Glycogen is a complex carbohydrate. Specifically, it is a polymer of glucose that is highly branched in structure, and it serves as a major form of energy storage in animals. These glucose molecules are linked together through alpha-1,4 glycosidic bonds, which creates the main linear chain and feature branch points that are connected via alpha-1,6 glycosidic bonds. This unique branching structure allows easy and quick glycogen utilization.
The structure also has a reducing end that is typically attached to the glycogenin, and a non-reducing end, the sites where enzymes can quickly add or remove glucose units. Lastly, the glycogen structure also has glucose monomers and branch points.
Going back, glycogen comes from the carbohydrate consumption. But how does this happen? This process is known as glycogenesis or glycogen synthesis. Glycogenesis is a metabolic pathway through which glucose, a primary sugar obtained from our food, is converted into glycogen. This occurs in the cytosol, the fluid part of our cell, where other metabolic processes takes place. Glycogen formation occurs when there is an excess sugar or energy from our body. After consuming particularly carbohydrates, our blood sugar levels rises. But in response, our body stores glucose in the form of glycogen.
Also, the location of glycogen storage plays a specific role. The glycogen stored in muscle cell is used exclusively to supply energy for muscles, while the glycogen stored in the liver is mobilized to maintain blood glucose levels and to support the energy needs of other tissues throughout the body. And this process is not spontaneous. It requires the energy in the form of ATP or adenosine triphosphate and UTP or uridine triphosphate, and as well as participation of multiple enzymes. Now, let's take a closer look at what happens in glycogenesis. To start the process, once inside the cell, glucose undergoes a series of transformations. First, glucose is phosphorylated by adding phosphate to the 6 carbon in the glucose using the enzyme hexokinase to form the glucose 6-phosphate, hexokinase 1 and 2 in muscle and hexokinase 4 in the liver. Next, the glucose 6-phosphate isomerizes to glucose 1-phosphate, and this reaction is caused by the reversible action of the enzyme phosphoglucomutase. Third, to activate glucose for glycogen synthesis, it involves adding UTP or uridine triphosphate to the glucose 1-phosphate to form the UDP glucose and pyrophosphate that is hydrolyzed by the enzyme UDP glucose pyrophosphorylase, which releases energy.
Next, UDP glucose acts as a glucose donor to the growing glycogen chain. The enzyme glycogen synthase is responsible for elongating the chain by adding glucose unit through alpha-1,4 glycosidic linkages. However, glycogen synthase cannot initiate the glycogenesis process on its own because it can only add glucose unit to an existing glycogen chain. Thus, it needs a pre-existing primer to bind to and extend, known as glycogenin. Glycogenin starts the chain by attaching a glucose unit from UDP glucose to a tyrosine residue within its own structure, releasing UDP. Glycogenin then adds 7 to 8 more glucose units in a similar manner, creating a short alpha-1,4 link chain. Once this chain is established, the glycogen synthase can now take over and continue elongating the glycogen molecule by sequentially adding more glucose residue from UDP glucose, forming additional alpha-1,4 linkages, and increasing the chain length by 1 glucose unit at a time. However, glycogen synthase cannot form alpha-1,6 glycosidic bond, which are necessary to create the branch structure of glycogen. The enzyme responsible for creating these branches is a glycosyltransferase, also known as a branching enzyme. It catalyzes the transfer of a block of glucose residue from the linear chain to the hydroxyl group on the sixth carbon of a glucose residue, forming an alpha-1,6 linkage and establishing a new branch point in glycogen molecule. Overall, the glucose 6-phosphate, being the main substrate, is converted into glucose 1-phosphate, necessary to form the intermediate product UDP glucose, which acts as an activated form of glucose and generates the main product glycogen. But what happens if the enzyme responsible for glycogen synthesis are missing or not functioning properly? The body's ability to store, regulate, and use energy is compromised, leading to several complications such as low blood sugar, liver damage, and muscle weakness. These malfunctions can give rise to a group of rare inherited metabolic disorder known as glycogen storage diseases. Now that we explored the process of glycogenesis, we have learned that glycogenesis is a metabolic pathway that utilizes energy such as ATP and UTP along with several enzymes to convert glucose into glycogen. This process is vital for maintaining energy balance, regulating blood sugar level, and supporting physical activity. But what happens when our body needs energy? Well, that's where glycogenolysis comes in.
Did you know that during emergencies, glycogenesis can be rapidly suppressed to prioritize glycogenolysis and energy release? This shift is driven by hormones like epinephrine, which signals our body to break down glycogen for immediate energy rather than storing it. Remember that during fight or flight situations, glycogenolysis is activated. Now that we have explored the first metabolic process, which is the glycogenesis, it's time to delve into its counterpart, the glycogenolysis. Glycogenolysis is the enzymatic process of breaking down glycogen into glucose to provide energy or maintain blood sugar levels during fasting or increased demands of energy. It occurs during starvation, when the body breaks down stored glycogen to release glucose for energy in the absence of food intake. This process takes place in our liver and muscle cells. You might be thinking, why those organs in particular? Well, it's because they have the necessary enzymes to perform glycogenolysis. We're talking about the glycogen phosphorylase, the debranching enzymes, and the phosphoglucomutase that are all present and ready to go in the liver and muscles.
Glycogenolysis. Let's break it down together, step by step, how glycogenolysis actually works. In the liver, glycogenolysis serves as a key mechanism for regulating blood glucose levels. When blood sugar levels drop, the liver breaks down its glycogen stores and releases glucose into our bloodstream, ensuring a continuous supply for tissues and organs, particularly in our brain. In skeletal muscles, glycogenolysis provides energy for contraction and physical activity. Muscle cells utilize their glycogen reserves to produce glucose 6-phosphate, which enters glycolysis to generate adenosine triphosphate, or what we know as the ATP, for immediate energy. Unlike the liver, muscle cells do not release glucose into the bloodstream as they like the enzyme glucose 6-phosphatase. Let us begin exploring the different steps of glycogenolysis as well as the enzymes involved in the process.
Step 1. Initiation. Glycogenolysis begins with the hormonal activation of the enzyme glycogen phosphorylase. It is triggered
by signals like glucagon in the liver or epinephrine in the muscles. With the help of inorganic phosphate, this enzyme cleaves glucose units from alpha-1,4 glycosidic bonds of the glycogen chain. This releases glucose 1-phosphate or the G1P, shown as the sky blue hexagon in the diagram. Glycogen phosphorylase continues cleaving until it reaches a branching point known as the limit dextrin. A limit dextrin is that glycogen phosphorylase can only cleave alpha-1,4 bonds until it is 3 or 4 glucose units away from an alpha-1,6 branch point. Step 2. Hormonal activation and debranching. At the branch point, glycogen phosphorylase can no longer proceed. Here, the glycogen debranching enzyme takes over. This enzyme has two activities. The first activity is that alpha-1,4 glucan transferase transfers 3 glucose residues, or the orange hexagons, from the nearby linear chain. The second activity is that amylo alpha-1,6 glucosidase breaks the remaining alpha-1,6 glycosidic bond, releasing free glucose molecule, or the purple hexagon. So this debranching allows glycogen phosphorylase to continue breaking down the newly linearized chain. Step 3. The conversion of G1P to G6P, or the glucose 1-phosphate to glucose 6-phosphate. The release of glucose 1-phosphate, or the G1P, is then converted into glucose 6-phosphate by the enzyme phosphoglucomutase. This involves shifting the phosphate group from carbon 1 to carbon 6, preparing it for its next destination depending on the tissue type. Step 4. Specific organ processes. In the liver, glucose 6-phosphatase converts glucose 6-phosphate into free glucose, which is then released into the bloodstream to maintain normal blood glucose levels, especially important during fasting. In the muscles, there is no glucose 6-phosphatase. Instead, the glucose 6-phosphate directly enters glycolysis to produce ATP, or the adenosine triphosphate, that supplies energy for muscles contractions and activity.
Let us now summarize everything we've covered so far. Glycogenolysis is the process of breaking down glycogen into glucose, which occurs primarily in our liver and muscle cells. The main substrate is glycogen, while the key enzymes involved are the following: glycogen phosphorylase, which breaks the alpha-1,6 glycosidic bonds, the debranching enzymes, which cleave the alpha-1,6 glycosidic bonds to release free glucose, and the phosphoglucomutase, which converts the glucose 1-phosphate into glucose 6-phosphate. The products of glycogenolysis are free glucose molecules that are released into the bloodstream, and glucose 6-phosphate, which is utilized in glycolysis. But what happens when glycogenolysis is impaired? Similar to glycogenesis, it can lead to complications such as hypoglycemia, energy deficiency, storage disorders, lactic acidosis, and neurological issues. To prevent such complications, we must maintain a balanced diet, regular physical activity, proper hydration, and monitor our blood sugar levels.